Hello and welcome to this Defra Knowledge Transfer e-learning course, Introduction to Stable Isotope Ratio Mass Spectrometry Applied to Food Authenticity Testing. The content of this e-learning course covers the following topics: what stable isotopes are and how they occur in nature, how isotope ratios are measured, how stable isotope analysis is applied to food authenticity testing, data interpretation and the importance of databases, current official uses in food-based testing, and finally a summary and a forward look. So, what is an isotope? Not all the atoms of an element are the same. The number of protons within an atom's nucleus determines the element. Nuclei also contain neutrons. The number of neutrons can vary between atoms containing the same number of protons. These are called isotopes, which means equal place. In the example, the nucleus contains 6 protons. Therefore, the atomic number is 6, and the element is carbon. The nucleus also contains 6 neutrons, and so we call this isotope of carbon, carbon 12. This is the most abundant stable isotope of carbon, comprising approximately 99% of all carbon atoms. An isotope of carbon that contains 6 protons but 7 neutrons is carbon 13, which is stable and still reacts in the same way as carbon 12, but it has a slightly higher mass. The presence of isotopes of an element affects the standard atomic weights found on the periodic table. These standard atomic weights are valid for all normal materials containing the element. For elements with only a single stable isotope, such as fluorine, the atomic weight is known very precisely. For elements with more than one stable isotope, e.g. strontium with 4 stable isotopes, the uncertainty in the standard atomic weight is much larger. This is because the abundance of strontium isotopes varies between samples of strontium. The chart showing the distribution of strontium isotopes varies between samples of strontium, and this variability contributes to the uncertainty in the standard atomic weight. For some elements, this natural variability in isotope abundance is so great that it dominates the uncertainty in standard atomic weight, which is highlighted by expressing the atomic weight as a range, shown here with the example of oxygen. The variance in abundances of isotopes is generally expressed in 3 ways. Firstly, the atom fraction, which is the amount of the isotope in question divided by the amount of all isotopes of the same element in the same sample. It can also be calculated from the number of atoms of the specific isotope divided by the total number of atoms of the element. The atom fraction of an isotope is also called the isotopic abundance. Secondly, the isotope ratio, which is the amount of one isotope of an element divided by another isotope of that element. Again, the isotope ratio could also be derived from numbers of atoms from the different isotopes. Lastly, the isotope delta value. This represents the isotope ratio of a sample relative to that in a reference. If an isotope delta is positive, then the sample contains more of the numerator isotope than the reference. If the delta is negative, then the sample contains less of the numerator isotope. The numerator, the number above the line, isotope, is usually the heavier of the 2 isotopes expressed in the ratio, e.g. 2H over 1H, 13C over 12C. The reference will have an isotope delta of 0 exactly by definition. To illustrate, Here are the abundances of carbon-12 and carbon-13 in a sample and reference. From these abundances, we can calculate the ratio of carbon-13 to carbon-12 within the reference as shown. We can do the same for the sample, which has a slightly lower atom fraction of carbon-13 and hence a higher atom fraction of carbon-12 and a slightly smaller carbon isotope ratio. From the two carbon isotope ratios, we can calculate the carbon isotope delta of the sample relative to the reference. This is a very small number, so it's usually expressed in per mil, per thousand. The delta can also be negative, and this is a common feature of food components relative to the carbon isotope reference material, which is a marine carbonate. Isotope fractionation is the process that causes variation in isotope abundance. Isotope fractionation is an important concept and is essentially the partitioning of the isotopes of an element between two coexisting phases in a natural system. A natural system could be a plant, soil, or the hydrological cycle. The two dominant natural fractionation processes that need consideration are kinetic isotope effects and equilibrium isotope effects. The kinetic isotope effect is a unidirectional process and is due to differing energy requirements of isotopes during a phase change. For example, during evaporation of water from a lake, it takes more energy to vaporize the higher mass isotope, so the water vapor will contain a relatively higher proportion of the lighter isotope in comparison to the liquid water. Examples include evaporation, diffusion, dissociation, and photosynthesis. Most biological reactions follow the kinetic isotope effect where, relatively, more of the heavier isotope remains in the substrate and the lighter isotope accumulates in the product, as less activation energy is required for the reaction. Equilibrium isotope effects are not quite unidirectional, but they are not complete reactions either. The effect is associated with physical processes, similar to kinetic isotope fractionation, but describe how the isotopes are distributed among different phases of a molecule's existence in chemical equilibrium. It's a type of mass-dependent isotope fractionation and relates to how the molecules interact through their chemical bonds in their surrounding environment. At an atomic level, it is related to the bond energies, with heavy isotopes having a lower ground state than bonds with light isotopes, and so there is a preferential reaction of the light isotopes as less energy is required. So, how do we measure isotope ratios or isotope delta values? Isotope ratio mass spectrometry, IRMS, is used to determine the isotope ratios of hydrogen, carbon, nitrogen, oxygen, and sulfur. IRMS uses a magnet to separate the ionized gas molecules that are introduced into the system. Detection is using dedicated collectors, Faraday cups, and can, for example, collect the ions of mass 44, 45, 46 for carbon dioxide simultaneously. The relative amount of each ion, isotope, is measured and ratios calculated. As IRMS instruments have a gas source, it is necessary to convert the element in question to the analyte gas. Generally, the sample and a carrier gas are introduced into a reactor which converts the target molecule to the analyte gas. Any other gaseous byproducts are trapped or separated from the gas of interest. For stable isotope analysis of carbon and nitrogen, an elemental analyzer is used to convert the sample into the analyte gases ready for introduction to the IRMS. The sample material is wrapped in a small tinfoil cup and dropped into a quartz tube maintained at 1050 Celsius containing chromium-3 oxide granules. A pulse of oxygen gas is added, which produces a flash combustion with the tin capsule. Organic material is converted into carbon dioxide, various oxides of nitrogen, water vapour, oxides of sulphur, and halide-containing byproducts. The latter are removed by silvered cobaltous cobaltic oxide in the reactor. The remaining gases are swept into a second quartz reactor containing copper wires maintained at 600 to 650 degrees C. Here, oxides of nitrogen are quantitatively reduced to nitrogen gas, and the excess oxygen from the combustion is trapped. In the next stage, the remaining gases, CO2, N2 and water vapour, pass through a trap to remove the water. Next, the carbon dioxide and nitrogen gases are separated, either by chromatographic methods as shown here, or using a gas-specific purge and trap system. These two gases then enter the ion source of the mass spectrometer sequentially, but sufficiently separated in time to allow measurement of both carbon and nitrogen isotope ratios during a single analysis. For analysis of hydrogen and oxygen isotopes, a different configuration of elemental analyzer is used. Only a single reactor containing a tube which is usually made from and packed with glassy carbon. For hydrogen isotope analyses, chromium chips may also be mixed into the glassy carbon packing of the reactor or used on their own in a ceramic tube. This time, sample material is enclosed within a silver capsule. It is dropped into the reactor at a much higher temperature, where hydrogen and oxygen within the sample material are converted into hydrogen and carbon monoxide gases by thermochemical conversion or pyrolysis. Nitrogen within the sample is converted into nitrogen gas. Metal chips, such as chromium, prevent the formation of other stable pyrolysis products, such as hydrogen cyanide, that would otherwise prevent quantitative conversion of sample hydrogen into hydrogen gas. Halides are removed by silver wool. As before, the two gases, hydrogen and carbon monoxide, are separated either by chromatography or purge and trap, and then introduced separately into the ion source of the IRMS. The isotope delta values obtained by IRMS require calibration to ensure that the delta values are relative to the same reference. There are materials internationally accepted as standards for isotope delta calibration. These have an isotope delta value of 0 by definition. For hydrogen, the internationally agreed reference standard is Vienna Standard Mean Ocean Water, VSMOW. VSMOW has hydrogen and oxygen isotopic abundances that mimic the average surface waters of the world's oceans. The hydrogen isotope delta scale has two primary reference materials with values assigned by consensus rather than calibration. VSMOW, which defines the scale 0, and a second water reference material, Standard Light Antarctic Precipitation, SLAP, which sets the span of the scale covering the range of natural meteoric water. While VSMOW and SLAP reference waters could be used to calibrate routine hydrogen isotope delta measurements, they are very expensive. Therefore, isotope ratio reference material producers calibrate other commercially available reference materials against VSMOW and SLAP. The further along the traceability chain from VSMOW and SLAP, the larger the measurement uncertainty. However, by measuring samples against materials that are traceable by a series of calibrations to VSMOW, we can be sure that our hydrogen isotope delta values are comparable and compatible with those reported in other laboratories. For isotope ratio analyses, it is beneficial to have reference materials that are matrix matched to samples where possible, according to the principle of identical treatment. A number of food matrix isotope ratio reference materials have recently been produced, which may be used for quality control, quality assurance, and potentially for calibration of isotope delta values. So, how are isotope ratios used in food authenticity determinations? The plot illustrated here shows the expected carbon isotope delta, bottom axis, and carbon-13 atom fraction, top axis, for different plants, animals, and classes of materials containing carbon. While atmospheric carbon dioxide has a carbon isotope delta of about -8 per mol, the various groups of plants exhibit more negative carbon isotope delta values and lower atom fractions of carbon-13. Plants obtain their carbon from atmospheric CO2 during photosynthesis, which is then subject to fractionation. In effect, plants discriminate against carbon-13 and therefore contain proportionately less of it than atmospheric carbon dioxide. Looking at the three distinct photosynthetic mechanisms, firstly, there are C3 plants that use the Calvin cycle during photosynthesis. This includes wheat, barley, many grasses, trees, and a large proportion of plant species. Then there are C4 plants that follow the Hatch and Slack photosynthetic pathways to fix carbon dioxide to make plant components. This is used by plants in hotter climates to reduce water loss by reducing stomatal opening times and discriminates less against carbon-13, leading to less negative isotope delta values. Examples of C4 plants include sugarcane and maize. The third photosynthetic mechanism is crassulation acid metabolism, or CAM. The CAM pathway is an intermediate pathway used by succulent plants in tropical regions such as pineapple and vanilla. Differences in fractionation of carbon isotopes between photosynthetic mechanisms may be applied to food authenticity. We can discriminate between sugars from C3 sugar beet and C4 sugar cane using carbon isotope ratios, even if the sugars are chemically identical. Carbon isotope ratios are generally preserved between plants and animals that consume the plants, and we can use these differences to distinguish corn-fed and wheat-fed chicken. Natural vanillin comes from a CAM plant, while synthetic vanillin is usually derived from petrochemicals that have a similar carbon isotope ratio to C3 plants, and so isotope analysis can be used to verify the origin of vanillin. In some instances, carbon isotopes may be used to uncover adulteration of foods. The classic example is adulteration of C3 honeys with C4 high fructose corn syrup. This technique is made powerful by comparison of the honey-sugar carbon isotope ratios to the same carbon isotope ratio within the protein fraction extracted from the honey. The protein fraction acts as an internal standard, and differences in carbon isotope ratio between protein and sugar within honey indicate adulteration. If there are differences in feeding regime between countries in terms of the C3-C4 balance in animal diets, then carbon isotopes may distinguish country of origin too. Here a plot of the carbon isotope delta of defatted beef protein from UK cattle, USA and Brazilian cattle. The UK beef generally displays a C3 carbon isotope delta, while the USA and Brazilian beef has a C4 signal. Here, carbon isotope analysis can provide an indication of geographical origin. Conventional farming uses fertilizers containing ammonium or nitrate as nitrogen sources. Here, the nitrogen is derived from atmospheric nitrogen. Atmospheric nitrogen has a delta value of 0 by international definition, and nitrogen in fertilizers is close to this. In organic farming, the source of nitrogen added to soil is animal manure, which is relatively enriched in nitrogen-15. Furthermore, during the manure maturation process, there is a preferential loss of the lighter isotopologues of ammonia containing nitrogen-14, additionally enriching the nitrogen-15 content of the manure. However, the nitrogen cycles in soil are complex and can make interpretation of the source of nitrogen used in agriculture extremely challenging. Nevertheless, stable nitrogen isotopes can provide useful information into about the fertilizers applied to plants, especially when production regime claims are made. Looking at sulfur, typically the carbon to sulfur ratio in living tissue is about 200 to 1. Plants take up sulfur as sulfate and reduce it to produce the amino acid cysteine. There is apparently no fractionation of sulfur isotopes in transport across cell membranes and in incorporation. But there is a fractionation of plus 0.5 to minus 4.5 per mil in the reduction process, referred to as assimilatory sulfate reduction. Sulfur, in the form of sulfate, can act as an oxidant and is utilized as such by sulfur-reducing bacteria, dissimilatory sulfate reduction, which plays an important role in biogeochemical cycles. A large fractionation of plus 5 to minus 46 per mol is associated with this process, which is the most significant fractionation of sulfur isotopes. Sedimentary sulfate typically has sigma 34S values of about plus 17 per mil, similar to the isotopic composition of sulfate in the oceans, around plus 20 per mil, while sedimentary sulfide has a sigma 34S of minus 18 per mil. Sulfur isotope ratios in sea spray sulfate can leave a characteristic marine signature on coastal geographical areas, for example, grassland close to the sea, and so can be a useful geographic tracer for crops grown or animals grazing in those areas. Isotope ratios of hydrogen and oxygen can also be used to obtain geographical information for food and other biological materials. Looking at hydrogen and noting that the primary source of all organic hydrogen is the hydrosphere, the hydrogen isotope delta of the surface water of the oceans is constant and is approximately 0 per mil. Recalling that evaporation of water is a fractionation process where lighter isotopes evaporate preferentially to heavier ones, the rainfall cycle causes a decrease in heavier isotopes. Reiteration of the cycle as distance from the oceans increase augments the differences, causing an isotopic gradient from coast to inland areas, and this is called the continental effect. Water that has passed through the meteorological cycle of evaporation, condensation and precipitation exhibits a systematic global geographical isotope variation dependent on latitude, altitude and distance from the ocean. The hydrogen isotope delta of precipitation, therefore, shows variability around the world. Oxygen isotopologues of water behave in a similar way to hydrogen isotopes, and a similar isotopic landscape or isoscape can be created for oxygen isotope deltas in precipitation. This gives rise to the spatial variability in hydrogen and oxygen stable isotopes when the water is incorporated into plant tissues through photosynthesis and animal tissues through ingestion. This is spatial variability we utilize for confirmation of country of origin labeling and PDO and PGI labeling claims in food production. An example is the analysis of water within fruit juices. If the juice is natural, then the hydrogen isotope delta of the precipitation where the fruit is grown will be reflected in the hydrogen isotope delta of water extracted from that fruit juice. Alternatively, juices that are dehydrated, shipped and rehydrated with water at destination may have differing isotopic compositions due to changes in the deltas from the provenance water and the water used for e-constitution. It is therefore sometimes possible to distinguish fruit juices labelled as from concentrate from those that are not. In this section, the need for databases and how to create them will be considered. The oxygen and hydrogen isoscapes of precipitation can be used to create a model to predict the isotope ratios where food samples have not been taken. The isoscapes for precipitation are then used to create further models based upon the fractionation expected for water within leaf stomata and create an isoscape for leaf water without measuring leaf water directly. Alternatively, we can take a substance that is expected to reflect hydrogen or oxygen isotope delta values for precipitation, measure the isotope ratios in samples obtained from known locations, and fit a model to predict isotope delta values for those of precipitation. This may be useful in geographical origin determinations. This latter approach requires samples of known origin or authenticity. Even without a modelling approach, it is possible to use databases of isotope ratios for known origin food samples for comparison to samples of unknown origin or suspected mislabeled origin. Such databases are of particular value when one stable isotope is insufficient to verify a country of origin label, e.g. differentiating between Scottish, Welsh and English beef. The carbon isotope ratios of British beef fall into a tight band which are different to Brazilian beef. However, the devolved nations all have their own protected beef brands such as Orkney, Scottish, English, Welsh beef and so on, which carbon isotopes cannot distinguish. To resolve this, a multivariate model is required, incorporating all of the light element stable isotope ratios as well as heavy isotope ratios and multi-element concentrations that markedly improves differentiation of the geographical origin of beef across the British Isles. The map pin locations show the locations of the farms where the cattle were reared. Samples of beef were collected and locations confirmed using information from cattle passports. These were subsequently used to populate the database with a stable isotope and trace element data. Once a database has been populated with data from authentic samples, including data from, e.g., trace element abundances, it can be challenged with samples of unknown origin or to verify origin claims. Various multivariate statistical approaches can be applied with the aim of grouping the authentic samples in the database into categories, e.g., country of origin, Canonymical discriminant analysis is used as a robust supervised pattern recognition technique that maximizes the separation between predefined groups, here English, Welsh and Scottish beef. An advantage of canonical discriminant analysis is that it allows us to see which variables play the most important role in achieving the separation of the predefined groups. We can see in the separation of Scottish versus the English and Welsh beef in the x-axis or first discriminant function that strontium and rubidium concentrations are important geological markers. In the second function or y-axis, the concentrations of zinc, samarium and sodium give the major separation between England and Wales. When creating such a database, it is important to include parameters or variables that are expected to vary between locations. The fractionation processes previously examined for hydrogen and oxygen isotope composition of precipitation varies systematically across the globe, with gradients from coast to inland areas and higher altitudes and latitudes. Different photosynthetic pathways leave characteristic carbon isotope signatures in plant materials used for cattle diets. Fertilizers, both synthetic or natural slurry, can impact on the nitrogen isotope composition of plant materials used in cattle diets. Sulfur isotope ratios from sea spray sulfate can leave a characteristic marine signature on areas of grassland close to the sea. Strontium 87/86 ratios of the bioavailable portion of the mineralized strontium derived from the bedrock can give us an indication of the underlying geology of areas of food production. Ultimately, the hope is that when all of this information is combined into a multivariate statistical model, it is possible to develop a unique fingerprint for a particular geographical region that can identify the origin of a food or verify a claim. Isotope ratio analysis is currently used for a variety of investigations into food authenticity and origin. A number of isotopic methods have been officially recognized for the detection of frauds, including mislabeling of several commodities. The table lists the current standard methods that are suitable to confirm the authenticity or declared origin of food and drink. These methods have usually been collaboratively tested and have standard method performance criteria. The majority are also referenced in International Codex Standard 234 for methods of analysis and sampling. For enforcement work, it is preferable to use officially recognized methods. It is also recommended that laboratories are accredited to ISO 17025 or equivalent. Finally, CEN Technical Committee 460 on Food Authenticity, Working Group 6, is dedicated to the review and validation of IRMS standard methods and will publish a new standard for carbon and nitrogen isotope analysis in bulk materials by Elemental Analyzer IRMS. A summary of the key points and suggestions for further reading is now provided. Stable isotope analysis gives an extra dimension of information that can't be obtained from chemical compositional analysis and can provide unequivocal evidence in food fraud analysis. However, stable isotope analysis, even when combined with trace element analysis and chemometrics, is not definitive. It is a comparative technique that often requires an extensive database composed of authentic and contemporary products that allows meaningful comparisons with suspect samples. It is important that authentic samples reflect variability in geographical origin, seasonal variation, permissible technological processing and varietal differences in raw materials. Consequently, it may require sophisticated mathematical models to deal with that data and build reliable prediction tools for fraud interpretation. These models must be regularly challenged with anonymized samples from outside the training set. Further resources on isotope ratio analysis in general or its application to food authenticity can be found in these publications. Both are freely available from the firm's network website. For stable isotope analysis of foods, the book edited by Leslie Chesson and Jim Carter on food forensics is an excellent resource, as is Modern Techniques for Food Authentication, edited by Darwen Sun. This eSeminar has been produced as part of the Joint Knowledge Transfer Framework for Food Standards and Food Safety Analysis, and which is funded by the Department for Environment, Food and Rural Affairs, the Government Chemist Programme for 2020 to 2023, the Department for Business, Energy and Industrial Strategy, the Food Standards Agency and Food Standards Scotland.